Thanks everyone for being here today. I'm going to talk to you about mass spec in the ARC that you can use yourself. But I'll start out by telling you a little bit more about who I am. I earned a Bachelor of Arts in Ecology from CU, and then went on to do a PhD in ocean science from UC Santa Cruz, where I studied marine natural products structure elucidation, so solving the structures of compounds with biomedical potential from different marine sources, where I did a whole lot of mass spec and NMR, and where I kind of got the fundamentals of what I use in my everyday here in the ARC. I went on and did a postdoc in soil biogeochemistry, and in 2014, I joined the ARC Mass Spec Lab. I still do research on the interaction of molecular structure and fate of organic molecules and how those interact with global change. But today we are going to talk about mass spectrometry. So we'll walk through some mass spec basics, what the systems are composed of and what we have here in the ARC. And then we'll kind of walk through instruments one by one and how you get self-service on them, starting with ICP-MS, which is led by Dr. Jackie Chaparro. Then we'll talk about some about our MALDI, which is led by Dr. Alyssa May, GCMS, led by Paul Matthews, and then the LCMS, which I lead along with Gwen Willits, who's a talented undergraduate who works in our lab. And I just want to give credit to all these folks up front because they helped make this talk. And so this is not just from me, but it's from all 5 of us, I would say. So we'll walk through those systems and then go over how you sign up to get training and then what kind of training opportunities we've currently got going on in the ARC. So to get started, we have here the components of a mass spectrometer, and they're all the same. There's lots of acronyms and combinations of what you put these things together, but basically you have to introduce a sample, you have to turn that sample into ions, and then you have to somehow filter, separate, analyze those ions, and detect them to create a mass spectrum. And
Claudia, I'm going to pause here. We're still stuck on your first slide, it appears.
Oh, okay. Let me try something different.
Oh, now we can see it. The who am I slide.
Okay, good. Oh, Paul says he can move my slides. That's pretty cool. I can do it like this. Okay, so there's the real quick thing about who I am. And okay, great. And now this is the intro I gave.
Yep.
Let's see. Looks like the animation. Oh, there we go. Okay, cool. Now you guys can see Jackie. Now you can see Paul. Is this true?
Yes.
Okay, great. And there's Gwen. Okay, so thanks for letting me go, Corey. I had no idea. So here we are again at the components of a mass spectrometer, where we have the sample inlet, ion source, and mass analyzer, and where we detect the ions. And for each one of these categories, there's a whole variety of ways that you can introduce a sample. So what we have, kind of give you an overview of what's possible, and then the things that are in bold are things we have here in the ARC that you can use yourself. So we've got GC, syringe infusion, and direct introduction of samples. We have a whole bunch of different types of ionization, electron, chemical, and electrospray ionization, as well as direct detect ionization sources, MALDI, which is matrix-assisted laser desorption, and DART, which is direct analysis in real time, as well as ICP ionization. We only have a couple of mass analyzers, both time of flight and quadrupole. We don't have any ion traps, magnetic sector, electric sector, but most of our mass spectrometers and most modern mass spectrometers are some type of tandem or hybrid version where there's multiple quadrupoles put together, TOF, QTOF, those types of things. So we're going to we'll walk through kind of these components as a group in terms of the sample inlet ion sources and mass analyzers. And then we've got, for detectors, both electron multipliers and photo multipliers that we'll talk about at the end of kind of the introduction on the components of a mass spec. Starting with sample introduction, we've got a GC system here. This is basically a gas chromatograph is basically an oven that has a column inside, and you introduce your sample, and sample separation is really driven by the boiling point of the molecules and some interaction of the molecules with the column. So they go through, it separates the mixture, and then in this case, our detector will be a mass spectrometer. We also have liquid chromatography systems where you have your mobile phase, our solvents up here, and our systems are all reverse phase chromatography. So your mobile phase is the polar phase, and that's introduced through a pump that travels through a sample compartment where your sample is injected, and then is passed over a chromatography column, a stationary phase that generally separates analytes on their polarity, but there's other types of chromatography as well. And then that comes out and goes into the mass spectrometer. We also have ways to just infuse samples directly into mass spectrometers. This is really useful in terms of, I can do this, great. In terms of samples that have really low sensitivity for mass spectrometry, you can infuse a sample over a long period of time to average enough spectra to see a signal. This is something that happens with, we have some gold nanoclusters that we've worked on with the Ackerson group that we have to introduce samples this way. And then the last type of sample introduction is direct introduction. So with our DART and MALDI sources, you basically introduce the sample directly, and you can do this by just dipping a glass rod in your sample. You can do this with solids or with things that are not soluble in other solvents that work with either GC or LC. So that's all of the sample introduction types. The ionization sources are a little more in depth. The sample introduction is pretty simple. So we'll start out with electron ionization or chemical ionization. This is a source that's commonly used for GCMS. And here you can see that you have a heater that's connected to a filament here. And this heater heats up the filament to incandescent. And then there's an electron beam that makes electrons transfer from the electron beam here to the electron collector here. And in the meantime, your sample analytes are coming through this system, interacting with that electron beam. And in the case of electron ionization, you have your analyte vapor coming from the GC in this gas inlet. So your analyte interacts with this electron beam, and the molecules are in the gas phase. They interact with the electron beam for a hard ionization that fragments the molecule and produces a radical cation, a positively charged ion that then goes into the mass spectrometer. And this magnetic field is introduced to make the path of those electrons longer so that they can, it moves in a helical shape so that it can interact with the analyte ions. Now, chemical ionization happens in basically the same way, except in the case of chemical ionization, now you have a reagent gas. I think we use methane on our GC systems. And so this is a softer ionization than the electron ionization. The electrons interact with the reagent gas, and then the reagent gas transfers the charge to the analyte stream. So in CI, you're going to get good intact molecular ions that are really helpful for confirming molecular weight. And this can be done in positive mode or negative mode. So you can generate positive or negative ions of CI. Whereas with the EI, you've got really nice fragmentation that can be very specific to different analytes that can help you identify molecular structures. This is, like I said, the front end of our GC systems, the front end of, or the detector for our LC systems. The ionization source is usually electrospray ionization. And the process of electrospray ionization is shown here, where you have your liquid sample coming in from your LC system. And this is there's both a voltage applied across this gradient as well as a heating gas, a drying gas. And so as the electrospray droplets are produced, the solution is being evaporated, and the charge being carried on the solvent basically comes down to this Rayleigh limit, where the repulsion from the like charges overcomes the electrostatic interaction of the droplet, and there's this Coulombic explosion that happens. It's like my favorite thing to say when I talk about mass spectrometry. And so it explodes into smaller droplets that just contain single analytes, and the solution continues to evaporate until you end up with naked charged analytes that then go into the mass spectrometer. So that's how ESI works. In MALDI, MALDI is really different from these other types of ionization because you need something to be cold crystallized with your sample in order for MALDI to work. So this purple glop here is the matrix, and the matrix is generally something, this is synapenic acid. This is an example of a matrix that you use. And so you have this laser that hits your sample plate that has your sample dried onto it that's co-crystallized with the matrix. And then the matrix molecules actually absorb all of that UV energy. So in these matrix molecules, you always see these highly conjugated systems, so they can absorb that energy from the laser. And then the excited matrix molecules transfer their energy to the analytes, initiating desorption of the analyte. And then the gas phase collisions between the matrix and the analyte cause the charge transfer, and you end up with ions that are then moved into the mass spectrometer through this pulsed ion extraction process, which basically lines everybody up at the bottom of the flight tube so that then they can do their race through time of flight. In our last two sources, DART and ICP-MS, or ICP, inductively coupled plasma, and direct analysis in real time. Both of these are using plasma to generate ions. So in the case of DART, there's this glow discharge that's created by applying a voltage between these electrodes in this tube containing, in our case, we use nitrogen here. And the metastable ions move through into this gap. There's a physical gap between the end of the DART source and the beginning of the mass spectrometer. And this is where you would place your sample. So you just hold your sample here, or you have a spot card, and the ionization is transferred from that charged nitrogen gas to your analytes that then they move into the mass spec. And ICP is similar in a way because it's also using a plasma. In this case, it's argon gas. Your sample is introduced through a nebulizer in liquid form, much like what it looks like for the ESI, except then when it hits this plasma, then the charge is transferred to the analytes, but then the analytes are actually disassociated into their constituent elements and analyzed that way. And we'll talk a little bit more about that when we talk about ICP-MS as a whole system. So that's an overview of all of the ionization sources that are available to you. And you might be wondering, how do I know which one I need to use? So I made this flowchart some time back, and this lives in our Microsoft Teams space, so you can find it there. But basically, you want to think in terms of molecular weight, polarity, stability, and what types of molecules you have in order to determine which kind of ionization source you want to use. So for things that are lower molecular weight, this would be under 800 or maybe even 600 Daltons. If you have something that is ionic or polar, this should work well with electrospray ionization, but not so if it's nonpolar, but it is volatile and thermally stable, then you could go to EI and CI and GCMS. And if it's a slightly larger molecule, then and it's ionic or polar, then you would use ESI. And if it's not, then you could use MALDI. For larger molecules, almost all the answers are MALDI. You can see here, no matter what happens, MALDI's for big molecules. But you can often do ESI with large molecules as well if they carry multiple charges. So this is a resource that's available to you on our team space, and you can look at that anytime to help you decide what type of ionization is going to work well for your analyte. Now that we've made things into ions, we have to talk for just a minute about what kind of ions we've made. So we know they're carrying a charge, either a positive charge or a negative charge. And you can think of what helps give them that charge as an adduct. So in this paper from Bloomer et al., they make your molecule a smiley face, and then your adduct is the type of hat it's wearing. So it's still the same molecule, just looks a little bit different depending on which hat it's wearing. And common adducts that are formed for LCMS at least are protonation, sodium adducts, potassium, ammonium in positive ion mode, and in negative ion mode you can lose protons or add potassium or sodium and lose 2 protons. And you can also have more than one charge. So you'll see here this is a plus 2H or minus 2H. So when we're thinking about the signal that we're actually getting from a mass spectrum, it's important to remember that it's not the molecular weight. It's some version of the molecular weight that you have to be able to interpret to understand what your molecule's mass is. And this is primarily for small molecules. This isn't really true for large molecules, but across databases that have mass spectrometry, most things are singly charged. So that's what this is showing here, either positively charged or negatively charged. Most of the molecules are showing up. And then there's a handful that have 2 or 3 or minus 2 or 3, and only very few that have a whole bunch. This is not true for proteins, say, but for small molecules, this is true. And what that looks like in a spectrum here is your mass to charge ratio. So you're not only seeing adducts, but you need to divide the mass by the number of charges. So for this molecule, this is 1035, the mass would be 1034. But if you add 2 protons, then it's half that. So you need to be able to interpret your spectra in terms of both what type of adduct and how many charges are showing up in the spectrum. Okay, so we made ions, we know about adducts, and now we need to analyze them. This is the fun part. This is showing a cartoon of how a quadrupole works, or what happens when a quadrupole is working. So a quadrupole can be used as a mass filter, where depending on the voltages that are applied, the currents that are applied, you can select specific ions only to come out of the quadrupole. And what's happening here is, in the case of a quadrupole, you've got two of the poles that are direct current, so they're not changing their current. And then you have 2 that are alternating current. They're going between positive and negative. And it's the shape of this electric field in the middle of the quadrupole that lets you focus these ions as they move through it. So you don't have to know anything about this. You just tell it, I want ions between 50 and 2000. And then the mass spec does all the work of adjusting these potentials for you to only see the ions that are between 50 and 2000. A quadrupole can also be very selective for only a single ion, if that's what you wanted to do, for different downstream applications. So that's a quadrupole. And then in the case of time of flight, it is exactly what it sounds like. The ions start at the bottom of a flight tube, and the little ones go fast, and the big ones go slow. So the time of flight for that blue one is the fastest, and then the red one is the slowest. And you can use a set of calibration ions that you know their masses and measure their flight times, and then use that relationship to tell you what the mass to charge ratio is of unknown molecules that you're trying to analyze. So we'll put these mass analyzers together in a number of different ways when we talk about the systems as a whole, but that's it for mass analyzers. And similarly simple are the idea of detectors. So these are electron multiplier tubes. And what you're seeing here is basically the same thing in both of these processes, where you have an electron that hits the detector, and this is all taking place in a vacuum. And then through secondary emission, a single electron can make 1 to 3 more electrons show up when electric potential is applied. And this moves, in the case of a continuous dynode, bouncing off these walls of this curve shape to multiply the number of electrons that are then converted to the signal for the spectrum that you're visualizing from the mass spec. And most mass specs today, instead of using direct continuous dinodes, use these discrete dinodes where there's essentially a phosphorus screen where the electron strikes to make a photon. And then the photon converts to a primary electron that then goes through this discrete set of plates where it multiplies the number of electrons and gives you the signal for your analyte showing up in your spectrum. So that's the whole thing. We did introduction sources, we did ionization sources, we did mass analyzers, and we did the idea of the detector. There's a couple more things to consider before we kind of talk through the systems that are available for your use in the ARC. And the first one is isotopes. I'm showing here 2 molecules of adenine, one that is just the most abundant version of all the isotopes. So protons, carbon-12, nitrogen 14. And in 1% of your molecules, one of those carbons is going to be a carbon-13. And this is something you can see in a mass spectrometer. You're discretely analyzing these individual molecules as they're moving through that system. And so the natural abundance of carbon-13 shows up in your spectrum. And you can actually use this to help you determine when you've got the correct molecular formula and when your signal makes sense for the analyte you're trying to analyze. So in this case, we've got an example of something that's 37 carbons, 68 hydrogens, a nitrogen, and 13 oxygens. And the molecular weight is 734 plus H, 733. And what's shown behind this in red and green are modeled isotopic distributions for predicted molecular formulas for this analyte. So you can see here the carbon, these are stacked. So the carbon-12 version of these molecules all look about the same abundance in their model distribution. And the red is the actual correct formula. And you can see when there's only 22 carbons, the height of that 13C peak, that M+1 peak, is below where you're expecting it to be if you actually had the correct molecular formula. So you use a combination of accurate mass and isotopic distribution to determine if you've got the right molecule, if you have a time of flight type instrument. And the reason this is relevant is when we think about, also, before we go to mass accuracy, it's also worth considering things that have really highly abundant natural isotopes. So chlorine and bromine are good examples. There's about a 33% natural abundance of the chlorine 37 isotope, and you can see this clearly in a mass spectrum for both when you have a single chlorine, and then if you have two chlorines, you've got a shape where you have two chlorine 35s, the 35 plus the 37, and then two 37s. And so this is all very modelable and knowable when you're looking at potential molecular formulas for compounds that you're interested in. The isotopes also play a role in understanding mass resolution and accuracy. So people say high resolution mass spectrometry all the time, and they may or may not know what they mean by that. So resolution is just the separation of your different signals in that spectrum. And most often what people are after is really high mass accuracy. But in order to get high mass accuracy, you need high resolution. So here I'm showing different types of mass analyzers. We talked about time of flight. Here you can see a typical resolving power might be around 10,000 full width at half max. I'll explain that in a second. And for something like an Orbitrap, it's 100,000, and for ion cyclotron resonance, it's up at 1 million. So what does that mean? What does that get you in terms of your spectrum? This is what it means. So this is a ion trap. The resolution is about 1700, and this is a molecule that weighs 868, I think. And you can see the separation between these peaks is not very big. This is that same molecule, but now this is on a high resolution TOF instrument. The resolution is about 50,000. And this is your carbon-13 peak, right? And 2 carbon-13s and 3 carbon-13s. So you can actually see the clear distance improvement, the resolution between those two types of mass analyzers. And when you get to an FTIC-RMS, not only can you see the difference between the carbon-12 and carbon-13 peak, you can also see resolution of the nitrogen-15 peak from the carbon-13 peak. So as you add resolving power, it's easier to hit what's the true apex of this peak for mass accuracy, and also to have fewer components that are contributing to where the middle of that peak is to get the best possible result from the system. So we do not have an FTIC-RMS, but we do have these high resolution TOFs and can do easily spectra that look something like this. All right, so that's all the background. That was like half the talk was the background, but now we're going to get to talk about the fun stuff with what types of systems you can use yourself in the ARC. We'll put all of these components together and talk about details of the systems themselves. So for the ICP-MS, you have this torch that atomizes and ionizes the samples, and you can see there's a drying phase and a dissociation phase where you get down to single elements that you're looking at. These ions turn a corner using a quadrupole, and then they go through this mass filtering quadrupole to a detector that counts the ions. And in ICP-MS, you're really focused on inorganic elements for the most part. And these are also really powerful. This instrument is really powerful in terms of its detection limits. So you can see the scale here is in parts per billion, parts per trillion. So sensitivity is really great. Pay attention though to the little dog ear color, not the main color, because we do not have our system in a class 100 clean room. It's just in a regular lab. So you're only going to get 1 to 10 parts per trillion instead of below 0.1 parts per trillion. So you're still doing pretty good. The organic elements don't show up as well because they're in everything. And so it's hard to analyze atoms of carbon in this system because there's so much carbon in everything around us, it wouldn't work very well. But all of these other elements are readily measurable by ICP-MS. And this is the domain of Dr. Jackie Chaparro. We have a Perkin-Elmer Nexion 350D. It lives in microbiology C110, and she runs training on this for users in 2 4-hour sessions. So there's a lot that goes into sample prep for the ICP-MS, and then there's a lot that goes into the data analysis. And so Jackie will run training sessions as needed for up to 4 people if you want to use ICP-MS. The system can only handle liquid samples that have less than 2% dissolved solids. And usually you're dealing with aqueous solvents that are between 2 and 3% nitric acid, just so you know a little bit about the sample prep for that system. ICP-MS is used across all the disciplines, I would say. We have users that apply this system for food testing, geochemistry, chemistry, archaeology, environmental studies, energy and fuels, and clinical research. So it's a really broadly applicable tool that works kind of differently than the rest of the mass spec we're going to talk about is really focused on organic molecules. So this is a good one to know about. The next system that we will talk through is the MALDI-TOF. So we already went over what a MALDI ion source does, the matrix-assisted laser desorption. And that's coupled to a time-of-flight mass analyzer and a detector. So the way this works is you mix your analyte with a matrix on a stainless steel plate. You desorb that sample using the UV laser, making ions in a gas phase. And then you accelerate into the flight tube using high voltage and measure the time of flight of the ions as they hit the detector and correlate that flight time to calibrators to measure your analytes. MALDI works really, really well for large molecules, so polymers, proteins, peptides, nucleic acids, and other biopolymers, as well as nanoparticles. But you can also use it for small molecules, especially when things aren't really soluble and things that work well with ESI. And our system is a TOF-TOF. So we have this sort of different arrangement than maybe what you're used to thinking about in terms of a tall flight tube sitting on top of a mass spectrometer. The Ultraflex, you'll see a picture on the next page. The flight tube is linear or axial, so it's on its side basically, which minimizes how the ions have to travel through the flight path and has some advantages. And there's 2 ways we can operate this system. So there's a linear detector, which is at the top of the flight tube. This is a lower resolution for high molecular weight compounds because for things that are big enough, they just can't make this turn. Alyssa has a great analogy where she talks about a bus on a mountain road. If you're going too fast, you're not going to make it around the corner and you're going to crash in off to the side. So for large molecules, like 500 kD, kilodaltons, you're going to use this linear detector. And for small molecules and for higher resolution, things that are less than 20 kilodaltons, you can use the reflector detector, where the ions move to the end of the flight tube and then they get this additional flight time that gives us the added resolution for this system. And then we can also do MS-MS with this system. So for things that are less than 5 kilodaltons, you got to run the reflector direct mode first to figure out what the MZ of the ion is that you're interested in. And then all the ions here are going to get fragmented after the source. And this pick selects the parent ion that you told it that you wanted to see from this reflector detector. And so that's the only one that comes out. And these are all the fragment ions that were part of that molecule originally, and then they separate in their time of flight, with the small ones arriving first and the large ones arriving last. So we have the ability to do MS-MS with this TOF-TOF setup for the Ultraflex. And this is what it looks like. It's like a big rectangle. But if you open up the top, you can see here is where your plate is introduced, and the time of flight tube is here sitting on its side. Alyssa runs this system. It lives in chemistry C5. She also does 2 4-hour training sessions, one on sample prep, because that's also critical to getting good data in MALDI, all mass spectrometry, but MALDI is really especially sensitive to determining what matrix you need and how to prep your samples. And then she does another about 4-hour training session on data analysis and acquisition. There's workstation and remote access to this system, and for your mass accuracy, you can get down to 5 ppm in that reflector mode and 60 ppm in the linear mode. Resolution is about 40,000 for reflector and 1200 in linear. This has low sample volume requirements, femtomolar sensitivity. You can use it with any solvent, and even solvent-free solids are possible. So that's the story about the MALDI. Applications with the MALDI that we've used it for, that we have users using it for. These are all kind of macromolecule type applications where you got oligonucleotide substitutions, polymer structural analysis, post-translational modifications, and macromolecule functionalization. So if you have any of those types of problems for your analysis, the MALDI is a great tool. The other thing that we're currently building for the UltraFlex is this BAC-ID method, where you take a protein spectra. So this is showing a dendrogram of relatedness of protein spectra generated from a MALDI-TOF, and comparing that to the phylogenetic relationship from all these different bacillus. So you can see that the phylogeny and the protein spectra match really nicely. This isn't always 1-to-1, but it's pretty close. And then you can couple that with a small molecule analysis for these individual strains and generate this molecular association network. So if we take, for example, this B001 bacillus, this is a colony that is literally like picked up off the plate, scribbled on the MALDI plate like a little Scantron with some matrix on top of it, and then you put it into the MALDI, you measure the proteins, you measure the small molecules, and you can see for this bacillus, these are all the shared metabolites, small molecules, with these other strains of bacillus, but these are all molecules that are unique to this strain. So if you're doing some kind of metabolic analysis, or you have a culture collection that you're curious about their metabolic potential and how they process different things, this is a really powerful tool that has this ID-BAC workflow that's being built into the GNPS2 platform that's in beta testing right now, and we're participating in the beta test for this to make it more widely available. So that'll be coming online soon. So that was MALDI. And now we're going to move into GCMS. And basically, like I already mentioned, you're separating compounds based on their boiling point and their interaction with the stationary phase, the GC column. You do direct liquid injection from the autosampler, or we also have thermal desorption available. So that's where you can concentrate gases onto a tube for things that are really low abundance in the atmosphere, and then analyze them all at once using the GCMS. All the vaporized compounds are focused on the head of the GC column and then separated through the column. And they are introduced into the ion source, where they have electron ionization or chemical ionization, depending on which source you need, filtered through a quadrupole and then detected. And these are super characteristic and super repeatable. You can compare your spectra with library databases to confirm molecular identities. So it's a really reliable way to assess known compounds. Paul Matthews oversees all the GCMS in the ARC. He does all the GCMS things. So I'm always like, I don't know, talk to Paul. So if this is in chemistry C5, there's a 1-hour training that you need to go through with Paul in order to use the instrument. And we have 3 nominal mass instruments plus a thermal desorption tube conditioner if you want to do TD. ArcBio also has a full-service GC accurate mass system. If you've got things that will only work with GC, but you really need accurate mass, that's something we can accommodate. So I just want to add that to our listing of self-service systems that are available, and these are all available for workstation and remote PC access. So this is an overview of the 3 systems we have that are available for self-use. The TSQ, the thermal desorption ISQ, and the Agilent. These are all helium with EI, and then the TSQ also has CI available. You can use, we have a triple quad on the TSQ, that's the T in TSQ. So that's really great if you need to do quantitative studies on different things. And then there's just components for individual features of these systems. And then this is that APGC TOF. This is basically chemical ionization as well, but you get accurate mass information from that system. Applications of our users that use these systems for GCMS are hydrocarbon and fuel characterization, carbohydrates, amino sugars, lipid biomarkers, fatty acid profiles, pesticides, and terpenes are all really common assays that users use on this system. And then there's, for thermal desorption, people are looking at atmospheric pollutants, off-gassing, and other kind of low concentration molecular weight volatile compounds in the atmosphere. You can do qualitative and quantitative analysis using standards and library matching, and formula and structure elucidation using accurate MSMS with the APGC TOF. So that is the GC story. And now for LCMS, we already kind of talked through this inlet method. This is Agilent. We have an Agilent 1260 UPLC system that is the sample introduction to, this is Agilent 6545 QTOF that has a quadrupole mass analyzer coupled with a hexapole collision cell and a TOF mass analyzer. Some kind of really updated features of this system is a wide bore lens 2 that keeps the optics really clean, so that there's increases sensitivity because things tend to stay cleaner with that wider bore lens. So this is the mass filter, the quadrupole, where you can control the mass of the ions that are coming through. This is the hexapole collision cell, where we use nitrogen gas as our collision gas to introduce fragmentation if you're interested. That's off if you want full MS and on if you want MSMS. The slicer design has options for high resolution or high sensitivity on this system, and then the pulsar design has better control for delay times to line up the ions here at the bottom of the flight tube to increase the stability of the mass accuracy. And then the power supplies are on this system are better thermally and environmentally regulated, which leads to better resolution of the spectra. So that's how the ion path in the QTOF LCMS system that we have. This is Gwen and me. We run this together. Gwen does training for folks that don't need chromatography if you just have a single sample that's already pretty pure. We have about a 1-hour training session that we have some pre-work with movies and an online quiz that you do before you come in and basically do a proficiency test with us, located in C5. And then I help folks out if you need to do chromatography for mixtures. We have this Agilent UPLC, but this is where we can also do syringe infusion or that dried spot or direct introduction using the DART source. And with this system, we are getting about 40,000 resolution with sub 2 ppm mass accuracy. And these are available for on a workstation and remote PC access for analysis as well. So what people typically want to do with this system is structure elucidation or compound confirmation. And so they're really interested in molecular formula prediction and unknown analysis. So this is an example. Say you have an analyte at 247 and you have all these possible molecular formulas that are maybe within your possible error that you might see on this system. These examples they all have halogens in them, so it'd be pretty easy to tell if you had a halogen because you would look at the isotopic distribution. But here we're just looking at the M plus H ion, and one of the problems people come up with unknown analysis is, well, if I can't use isotopic distribution, if everything's just chlorine, nitrogen, hydrogen, phosphorus, what do I do? And this is something that happens. So for example, if you had a mass that was 570, an M over Z that's 570, and you search that in a mass spec database, for just adducts that are M plus H, you might end up with 162 options of what molecule that might be. And you may want to narrow down that unknown to something smaller. And if you don't limit the number of adducts that you've got and include everything that's here that you can select for your database, you come up with almost 1,400 hits. And so what do you do when you don't know which one of these things that you're working with? And there's some automated solutions. Corey has come up with RamClustar is a really awesome automated processing workflow that will take your spectra and analyze all of the ions that are present and tell you when you've got an M plus H and M plus sodium. And I think in a lot of ways it outperforms some of the other algorithms that do this. Here you've got the same spectra, but now this algorithm is calling this M plus H minus water minus CH2O2. And so you can see like this is a tricky interpretation problem. And we kind of end up in this bag of, we've got our known knowns, things that are in databases and in mass spec databases and just compound databases. So like KEGG has about 18,000 compounds. And you've got your known unknowns. So you could imagine if you have a cyclic tetrapeptide, something with 4 amino acids, you can draw 40,000 possible structures, and 65 of those are known. So you know those unknowns, and you could predict what they might be. But what about this kind of like Rumsfeldian space of the unknown unknowns? How do you know what you don't know? And is this really a problem that we need to think about in terms of molecules and mass spectrometry? And the answer is very much yes. So this is sort of like a fun little exercise in the chemical space occupied by molecules. This is for just the chemical class of enamines. And this is sort of like possible space where there's maybe 4 billion different compounds you can imagine that are enamines. And for reference, the estimated number of stars in the Milky Way is 1.7 billion. So we're talking about like hard numbers to even think about how many different possibilities are there. And then there's some like kind of computationally generated databases of things that might be more likely to be possible. And then here there's still 96 million of these things in PubChem, right, that are real. And so if you have this functionality, how are you supposed to figure this out? And I lean towards thinking about natural products and unknown analysis because that's what my background is in, particularly for microbes. And even for these different species of actinobacteria, people have studied, how many different types of natural products. These are a lot of our antibiotics come from these molecules. And are we likely to find new antibiotics from these organisms? And estimates are basically, depending on their genome size and the number of analytes we've already identified from them, that there's still like 70 to 90% of their chemistry is yet to be discovered. So there's really like a lot of unknown unknowns out there. And how do we solve this problem with mass spectrometry? So one strategy to do this is this, I mentioned earlier, this GNPS, which is Global Natural Products Social Molecular Networking. This is just a fancy way of saying that you're going to look at your MS spectra, your fragmentation patterns, and group molecules together based on what their classes are. And this uses a workflow called classifier, which automates their structural classification based on something like kingdom, class, families, genus, species. So it's an analog for that, but with molecules. And so there's about 5,000 different classes of chemicals that you can characterize using GNPS. And then you can delve down into these molecular networks and look at what structures are shared in common based on the fragmentation pattern, and make some of these unknown unknowns, turn them into knowns so that you can continue on with your research. And so this is something where we've built kind of a training course around that I'll talk about at the end, but first I wanted to introduce, just mention quickly, that's kind of gives you an overview of the instruments and applications that we have here available to users. And the way that you go about getting trained to use these systems is to join our team. So we've got in Microsoft Teams an ARC users team, and there's a Mass Spec Lab channel in there with a self-service notebook that has basically everything you need to know in order to use any of the mass spectrometers in the ARC. So you can join that. You can email me and ask to join that. We can invite you, whatever works the best for you. And then once you're in the MS team, the way you request training is through our iLab site. This is something that works with your CSU EID, and you can sign up to get trained on any of the instruments that we talked about today and tell us a little bit about what your samples are and what you're trying to do, and then you'll get scheduled with the right person, Jackie or Paul or me or Gwen or Alyssa, in order to use the instrument that you want to use. And I'll just wrap up with a short plug for our metabolomics self-service training course. We were planning to offer this earlier in the year, but we've switched the analytical platform to that Agilent 6545 QTOF instrument. So we had some optimization to do for that. But if you're interested in learning about metabolomics and unknown analysis, we walk through, there's about four 4-hour sessions over 2 weeks, where we walk through sample preparation, data acquisition, processing and analysis, and visualization. And if you want to participate in that, can sign up through this QR code, takes you to an interest form for that, or this is the link to that forum, or you can just email me and I can send you these things. And I think that is all I had to share today. So thanks for coming, and I'm happy to answer any questions. Jacob?
Hi, Claudia. Great talk. This is a fairly naive question, but like, what's the process for ARC acquiring like a new instrument? What's that process look like? And do you guys have any intentions of acquiring any new instruments? You guys have a fleet of lots of different instruments that can analyze different molecules and such. I'm just kind of curious if there's any intentions on acquiring a new instrumental setup.
We're always thinking about that. That's a great question. Is there a particular instrument you'd like to see in the ARC?
Oh no, I'm just curious in general.
Curious.
And how
Okay.
do you ever call it's like, okay, we've got the instruments that we need, and like, how does that process
No,
work?
we've never said that. We always want more instruments. But it's really driven by the researchers. So if there's people that have questions they can't answer with the platforms that we have, we try and point them in the right direction. And if there's enough interest in upgrading or adding capabilities, then we've got a few different routes to do that. So instrumentation grants are one. We are always submitting instrumentation grants to NSF and NIH programs we have ones pending for, I know we have 2 NMR ones pending, as well as an XPS one. And we have tried to fund a couple times a new QTOF through that mechanism, and we were unsuccessful with those. And so then we got together with the VPR and PIs and the colleges basically, and people can pool resources to get something that they really want to have their researchers have access to. So there's a number of different mechanisms, and we basically have a list, like a wish list. There's stuff, that we know would benefit researchers on campus, but if there's something that you're thinking of or your PI is thinking of that you're like, it'd be really great, then definitely let us know, and then we can, kind of prioritize that and see how we can identify funding to bring that. I would say startup packages are another place that we can kind of work with people, where if there's a new PI coming to CSU and they have startup and they want to have some type of equipment that would benefit multiple people, then sometimes they're willing to put that equipment in a core facility so that other people have access to it too.
Awesome. Thanks so much. And I appreciate the plug for the metabolomics training course. Seems pretty cool. I'll definitely plan on checking that out.
Okay, cool.